Hi guys, Miss Francis here to discuss mitosis with you. So today's vocabulary is mitosis, and you must understand the 5 phases of mitosis and what occurs during each phase by the end of this video. So PMAT is the acronym that we use to remember the 5 stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. And remember that interphase is occurring before mitosis occurs, and then cytokinesis happens once mitosis. These 5 stages are done. So here's interphase right before mitosis has started, during the late G2 phase of interphase. You've got your centrosomes with 2 centrioles. That would only be found in animal cells, where you're beginning to organize the microtubules into an aster or a star. So this here is an aster. You've got a cell membrane, but you've also got a nuclear membrane present. You have a nucleolus, and your DNA has already been duplicated. That happened during the S phase of interphase. The chromosomes are starting to coil, they're becoming condensed, and then they're going to be visible during prophase. You'll need to know how to identify the various phases in onion root tips. Can you see how these 2 images inside the red box depict an onion root cell tip in interphase? Then we have early prophase, or just prophase, and we're going to start with a chromosome number that's 4. This is known as the diploid number. We're going to get into something called the haploid number eventually, which is half the number of chromosomes. The diploid number is the full number of chromosomes, and this particular cell has got 1, 2, 3, 4 chromosomes. So we're going to trace those chromosomes throughout mitosis. So during early prophase, or just prophase, you've got the mitotic spindle starting to form, which pushes the centrosomes towards the poles. You've got centrioles only present in animal cells. The nuclear membrane begins to disappear. Your sister chromatids join together. You don't have a nucleolus anymore. That nucleolus is gone, and chromosomes are now tightly coiled. How can you tell that this cell is in prophase? Because you can see the linear chromosomes starting to form. Late prophase is what we now call prometaphase. In some textbooks, you'll see it's still written as late prophase, but we're going to call it prometaphase. So now you've got fully formed spindle fibers. The nuclear envelope breaks down. In other words, the nuclear envelope disappears. Your chromosomes are fully condensed, and they are randomly dispersed throughout the cell. Microtubules from one pole attached to one of the 2 kinetochores. So microtubules are a special kind of spindle fiber. Kinetochores attach to the centromere and eventually breaks apart the sister chromatids. That's what the microtubules do. So let me get into the specifics of spindle anatomy. Right, so here's the aster, the star-shaped collection of the centrioles. Here's a kinetochore microtubule. The way we know that it's a kinetochore is because it's attached at the centromere. So kinetochores are proteins attached to the centromere. Here would be a non-kinetochore microtubule because there's no centromere that spindle fiber or microtubule is attached to. So a microtubule is same as a spindle fiber. You can have 2 different types. You can have a kinetochore microtubule or a non-kinetochore microtubule. So notice that chromosomes are more pronounced in pro-metaphase versus prophase. And then we're going to head towards metaphase. So let's explore metaphase. Metaphase, you've got your sister chromatids lining down the metaphase plate, which is also known as the middle of the cell or the equator. Can you say that we still have 1, 2, 3, 4 chromosomes that are replicated? They still have their sister chromatids attached by a centromere. And you'll see we've got kinetochore as well as non-kinetochore fibers. In anaphase, our microtubules, also known as our spindle fibers, push the poles apart. And what that does is divides the sister chromatids. So the kinetochore microtubules pull the chromosomes towards the poles. The centromeres divide, separating the sister chromatids evenly. So we still have 1, 2, 3, 4 chromosomes that are going towards this pole, and 1, 2, 3, 4 chromosomes that are headed towards this pole. So now the two poles have equivalent collections or equivalent clusters of chromosomes. Which cell is in anaphase? How would we know? Well, can you see that this cell would be in anaphase because our sister chromatids are pulling apart towards either end of our cell, heading towards opposite poles. Lastly, we've got telophase. In telophase, the spindle disappears, the nuclear membrane reforms, the nucleolus reappears, and those chromosomes start to decondense. In other words, they start to uncoil. But do you see that we have 1, 2, 3, 4 chromosomes in this top cell, and we've also got 1, 2, 3, 4 chromosomes in this bottom cell. This is an animal cell. So you're going to see something called cleavage furrow where the membrane pinches in. And then our last stage, So how can we differentiate between telophase and anaphase? Well, can you see that this would be anaphase can you see that this would be telophase, where your chromosomes have headed towards the opposite poles? Here would be anaphase, where our chromosomes are starting to be pulled apart. Here we've got metaphase, here we've got interphase, and here we've got prophase. So lastly, cytokinesis is the division of the cytoplasm. So once mitosis is over, cytokinesis can take place, but cytokinesis is slightly different dependent if this mitosis is occurring in an animal cell versus the plant cell. So in animal cells, you'll have a cleavage furrow forming, which includes an actin ring, whereas in a plant cell, what you form is your plate cell, and it gives rise to cell walls. Then, once interphase is over, we've got our identical daughter cells. Our chromosomes are fully decondensed, and we are now back in early G1 or early gap 1 of interphase. So what's different about metaphase in both of these images? We see that the chromosomes are lined up down the middle, down the equator, but how do we know that they're different? Well, do you notice the shape of this cell versus the shape of this cell? So what we're looking at over here is an animal cell in metaphase versus on the right-hand side, which is a plant cell in metaphase. There are 2 videos that you can revisit to go over the stages of mitosis. There is Mitosis Made Super Easy and the Stephanie Castle video as well.